Well, good morning, students and friends and colleagues and everyone else who's listening. Hope you're having a nice morning. Hope that your fall is going well and you're settling into classes and everything's going the way it should. My streaming setup is back online. My computer's back from the Apple Store, so I'm running at full capacity here once again. New background, but don't adjust your TV sets, folks. It's just a rearranged office. Anyway, so today we're going to talk a little bit about the concept of developmental programming, which is our entry into the, I guess, kind of a bit more of the practical implications of the work we're doing, getting away from some of the background and kind of moving into some of the ways that the techniques and concepts we're talking about can can be used in sort of real practice. So without any further delay, let's go ahead and just start looking into it. Before we begin, I want to talk about the first midterm. So that's coming up on Monday, so a week from today. Now, I can't speak about it in overly concrete terms, partly out of, partly in interest of keeping it confidential and maintaining the integrity of the examination process, but also just because I haven't yet made it, so I have to decide what I'm going to put on it. But I want to sort of set out some parameters. Most of these you could see on the syllabus. The exam is going to be about 1.5 hours long. It's going to be, approximately the length of one class because I'm not holding a class that day. You will have the whole 24-hour period to choose, that unit of time and do the exam. It's going to be available between 8 a.m. on Monday to 8 a.m. on Tuesday. So it's a full 24-hour window. You can write your exam at any point in that window. So that's entirely up to you when you'd like to do that. The exam will be written on Brightspace. Once it goes live, you'll see a link on the Brightspace page that you can click, and it'll bring you to the exam. You probably, I'm sure you're probably used to this at some point, or by now, the idea of writing an exam online. So typical thing where the questions will be presented in random order. Where possible, the answers will also be randomized. You'll be able to go back and forward during the exam between questions. Obviously, that's something you could do if you had the exam in real life on paper, so I would certainly want you to be able to do that on the thing. It's going to be worth about 20% of the course grade, so a fairly valuable exam, and some fairly challenging content. I think I wouldn't expect that this exam is going to be a piece of cake for anybody. Hopefully, it'll be a good challenge that kind of adequately tests your knowledge of the material we've talked about up until now, in September 6th, up until the lecture on Wednesday, whatever the next lecture is. The format is going to be something like this. Multiple choice will be between 10 to 15 questions of the multiple choice variety, just kind of appetizers to get you in the mood for writing the actual exam. And then there'll be maybe 10 to 15 short answer questions depending on what the questions are and how long an answer that I want. So some of these are going to be short where you might need to write a couple of sentences in, 1 or 2 points kind of thing. Others you might have to write out a paragraph or more. So one of the kind of ways I like to ask a question is a fairly open-ended one where it's like, okay, so, explain how next generation sequencing works, or explain the role of hybridization in next generation sequencing, kinds of things like that, where I'm trying to, ask you to kind of lay out an entire concept in a narrative way that you can explain using your own words. I think that's a fairly good way of testing people on things. So it'd be something along those lines. Sometimes I might ask you to interpret a figure depending on what my mood is. So, figures of the sort that we've seen up till this point, I guess that's actually, probably that kind of question is going to be a bit more common in the second midterm and the final, when we've actually started dealing with more data and more figures that you would actually be interpreting regularly. But something along those lines might also come up. So I don't want to give too many more details now. I think that should give you at least a sense of what to study for. And obviously, if you have any questions, you can ask You can ask this week in the next class, or you could ask in office hours, or of course you can email me, or anything else that you might. There's a number of ways to reach me depending on what your preferences are. So let's leap into the material and start talking about the developmental origins of health and disease. So we are looking in this course at the genetic and epigenetic basis of brain development and mental illness. And we're going to have a lot to say about both of those topics. There's certainly a sizable genetic role in your risk of mental illness and various other things. However, as I think I might have explained in a previous lecture, even some very extreme conditions that seem to have a very penetrant phenotype cannot be explained entirely by genetics. So even for things like schizophrenia, if you have identical twins, it's not guaranteed that if one twin is diagnosed with schizophrenia that the other one will also be diagnosed. And so even for things that you'd think are so disruptive to life that they must be genetic, those are still not completely genetic. And there are some that are completely genetic, but many of them aren't. And so if you're looking for what's the missing piece if it's not genetics, well, it has to be some aspect of the environment. And if it's some aspect of the environment, you kind of want to think about when, if there's going to be a difference in the environment that predisposes you or gives you some kind of risk, when would that difference happen to create the maximum effect? If I eat some bad eggs for breakfast, I'm going to have kind of a bad day potentially if they're upsetting to my stomach or whatever, but it's not going to have a long-term effect on me. It's just a minor thing that, part of the ups and downs of everyday life. If I get a parking ticket or if I stub my toe getting out of bed, all these things are minor things that can happen within the scope of a normal day that don't have a long-term impact. And that would describe most things, I guess, if you think about it. Most things don't have a long-term impact. And so a lot of the ups and downs of the environment can't, I guess, plausibly cause these lifelong mental illnesses that we're going to be focusing on. On the other hand, if you have something happen during development, during kind of critical windows when your brain and your body are developing, even relatively small differences can have a long-lasting effect because they're happening while your brain is in sort of a more plastic and potentially more malleable state. So this is kind of what we're interested in with the field of developmental origins of health and disease. You can call this DOHAD for short, developmental origins of health, and then the A is lowercase and disease. So DOHAD. We're interested in the effect of early life environmental conditions on the risk of disease. And the classic example of this that kind of got the whole field going was looking at the cohort exposed to the Dutch hunger winter of 1944. So this was an event that took place in World War II. Perhaps you've heard of this war. It was a pretty big one that happened in the entire world. The Nazis had essentially blockaded the Netherlands and led to cities there, basically going without food shipments for an entire winter. Now, this required progressively harder and harder, or progressively more aggressive rationing steps, to the point where people were getting maybe 500 to 1,000 calories per day if they were lucky. Roughly about half the amount of calories you'd need to live a normal life where you're kind of in a neutral energy balance. And this is bad enough if you're just a laborer or a professor or something. But it's extremely bad if you're pregnant, because not only do you have to support your own nutritional needs, you also have to support the needs of your growing fetus. So as it turns out, if you follow people who had been in utero during this famine, if you look at them when they grow up to be adults, what you find is that they have higher rates of glucose intolerance. Glucose intolerance is sort of a precursor to type 2 diabetes, when you take some glucose or you eat something with sugar in it, and your body has a difficult time bringing your blood sugar levels back down to kind of a baseline where they belong. And females who were exposed during the earlier stages of gestation were also obesity prone. See, the beauty of this is it's a natural experiment. You could never, do this in real life as an experiment, because it would involve horrendous cruelty to human beings. However, if it happens to take place because of a war, for example, and in this case it was the Nazis that did it, and I don't think anyone's going to be apologizing for them. The experiment happened, and you can study the people who were exposed to it in a way that benefits science without having you having you be culpable for what actually happened. But anyway, so this whole observation that early life conditions of famine or stress and these kinds of traumas can predispose you to worse health outcomes has kind of morphed into this field known as developmental programming. Developmental programming, to define it, really just refers to changes in structure or function of a biological system that are caused by exposure to a stimulus in early life, where the effects of that exposure persist long beyond the duration of the initial exposure. So the Dutch hunger winter was, a winter. It took, several months to kind of work its way through. People who were exposed while they were in utero, fetuses, they were exposed for a very tiny proportion of their whole life. You're only a fetus for 9 months or something. That's not a very big percentage of your entire life. Yet the exposure that you receive here has such a long-lasting impact on your health and your future life course. So there's a lot of things you could look at in this context that would be potentially relevant to developmental programming. You can think about things like differences in nutrition, i.e. famine, stress, for example, natural disasters or war trauma and that type of thing. You can have infections like influenza or COVID. You can have a variety of different traumas that you could be exposed to during these early periods in life. See, the development of the brain is a very sensitive process. And if you've studied, developmental biology or the brain development courses here, it's almost astonishing that it's possible at all that the brain can kind of self-assemble with nothing more than some dividing cells and some properly organized scaffolding, both chemically and physically. And really, in order for it to go off well, it depends on everything being nicely timed and everything happening at the right time and in the right place. So, and it plays out, according to a fairly stereotyped pattern. The same developmental things happen in the brain at the same time, roughly speaking, for most people. So, of course, the first kind of few, the prenatal period is one where neurogenesis predominates. The postnatal period is one where gliogenesis predominates, and you have synaptic pruning, and you have apoptosis of neurons that are not needed or not part of the overall part of influential circuits. You have myelination. These things all happen at specific times, and because of this, there are what we call sensitive windows or sensitive periods where things that environmental exposures that happen during this time upset a very specific part of the developmental process, leading to long-term changes in that part specifically. So the timing is really what matters when it comes to exposure. Being exposed, for example, to famine in utero is much, much worse for you than being exposed when you're, say, 5 years old. It's not great when you're 5 years old, but you'll most likely recover. But if it happens to you while you're much younger and your brain is still in an earlier stage of development, the effects can be permanent, not just transient. So we look in mice a lot for this, and mice are in some ways simpler than humans. You can time mice, you can time the development of mice and divide it roughly into early or late prenatal and then early or late postnatal. The prenatal period for mice is about 20 days, kind of just split down the middle, between day, say 0 and 12 would be early, and then the rest of the days are late. And the same would go for the postnatal period. Mice begin eating solid food around day 14, I think. They have about a 21-day nursing or suckling period. So you kind of just split it down the middle. In humans, of course, we talk of trimesters when you're looking at prenatal humans and months or years postnatally. So you can distinguish between effects that happen during the first trimester versus effects that happen within the third trimester and so forth. And this was all kind of enunciated by this thing called the Barker hypothesis. There was a scientist named Barker who came up with this. The argument that he had was that this developmental programming phenomenon, this tendency for development to play out in a plastic way that's sensitive to the environment, is something of an adaptation that helps the adult organism develop in a way that suits the environment. So take, for example, the hunger winter. Now, we're equipped with genes that regulate our energy balance and our food intake, and the thinking would be that we should always try to eat as much food as possible and store as much energy as we can. But that, the relationship we would have with food and energy balance depends a lot on what's available in the environment. You might live in an environment where food is readily available, like the one we have, where you can, pretty much have as many calories as you'd like. Maybe not good calories, but calories all the same. And that's one thing. But you might also live in a very impoverished environment where calories are fairly scarce, and it's difficult to find all of the nutrition that you need on a daily basis. And our genes kind of don't know what environment we're going to be born in, because we could be born anywhere at any time. Your parents might have enjoyed a lot of, ready access to food, but evolutionarily speaking, maybe your generation happened to occur during a drought, no fault of your own, but there was a drought, and you're not going to have nearly as much food. So the logic would be that by making the development of the brain and energy balance systems here in particular plastic, would allow the developing organism to adapt its metabolism to suit the way the environment actually is. So, as an example, fetuses that were exposed to famine during the Dutch hunger winter might be programmed to be quote unquote thrifty, meaning that they try to save energy a lot. If you're thrifty with money, you don't try to spend very much of it, you try to save it. They would be given slower metabolisms and a higher tendency to store fat. And if you are growing up in a world where famine is the norm, this would probably be quite ideal. Now, of course, these developing fetuses did not know that the hunger winter was a temporary thing and things were going to go back to normal fairly soon afterward. So suddenly this adaptation towards saving energy and storing fat that they made in response to this famine is now completely wrong for the environment they're actually going to live in, where, in the Netherlands post-World War II, fairly rich country, they were able to afford all the food they would, need, and there's no need to have this adaptation. So what you end up with then is obesity and diabetes. This, anyway, is the argument of the Barker hypothesis, and I don't think that it's necessarily correct, but it's a very kind of appealing thought, I suppose, to believe that the development is clever enough to play out in this way that's sensitive to the environment. So the Dutch hunger winter is an example of what we would call a birth cohort study. As I was saying, experiments of this sort in humans are not possible to do naturally, but history is full of awful things, and if you wait around long enough, it's bound to be the case that something horrible is going to happen, and you can, as a scientist, you can come along and take a look at, or you can study those people and find out what's happening without ever being, responsible for doing the thing yourself. So the hunger winter was one of those, and then another example comes from China. This is from the Wuhu. Wuhu is actually a city in the I think the southeast bank of the Yangtze River. But anyway, it's part of China, and it just happened to be a place that these researchers studied. During the Great Leap Forward, there was sort of an attempt to modernize the economy and the culture of China, and it didn't really go terribly well. As a result, famine was widespread for a number of years. So what researchers found when they studied adults who were born during this time is that they have a significantly elevated risk of schizophrenia. So let's take a look at this figure and try to understand what we're talking about here. So this comes from St. Clair et al. In 1995 when they did this study. But here on the x-axis we have years, so 1956, 57, 58, etc., all the way up to 1965. On the y-axis we have births. One scale is births, ranging from 10,000 to 80,000. And then the other y-axis is the relative risk of schizophrenia, with the normal frequency in the population being about 1%, and the scale goes all the way up to 2%. So this is a twofold increase in risk if you happen to be, like I said, the average is about 1%. If you get up to 2%, it's a twofold increase in risk. And then these y-axes correspond to these specific lines. So the births are plotted against the births axis, and what you will see is that at the outset of this great leap forward that happened, between kind of like the late 50s and early 60s, births plummeted in the region. It's not easy to give birth to live babies when you're in the middle of a famine. So the rate of births went down. And at the same time, the odds of an adult born during this time of developing schizophrenia were increased up to twofold. And you can see that the two things are in wonderfully lined up, or directly out of phase. So as the birth rate drops, the rate of schizophrenia among babies who were born during this time goes up markedly, up to like double the average, or kind of double the regular risk. So what this shows is, and this is sort of just one kind of practical example of the type of cohort studies that you can do, is the trauma, the famine that led to all this stress and all this decline in birth rate also seemed to have an effect even on the babies who did survive, because their risk of developing schizophrenia was about twice as high as anybody else's. Now, I guess if you want to be saucy, you could argue that because this was a famine, what it maybe shows is not that this increased the rate of schizophrenia, but just that infants who were predisposed to schizophrenia had an easier time surviving. And so they had a slightly higher neonatal survival rate. And this is why you see more of them, or why they seem to be overrepresented here. I think that's probably not likely, but it's something you have to kind of consider. There's always another interpretation for the data. And yeah, so this is one example of a birth cohort study, and there are many such cases. Now, I think coming from our kind of position as neuroscientists and our status in the neuroscience department, it's kind of hard to avoid the fact that for most cases, while you might say you're studying starvation and infection and these kinds of things, at the same time, inevitably, stress is going to be happening as well. You can't imagine a scenario where a mother is starving but not stressed. Or at least it's very tough to imagine what her mindset would be if that were the case. So most of the environmental insults that we look at, whether it's infection or whether it's famine or anything else, also happen to cause stress. And stress is always kind of mixed and mingled in with these stories. And of course, the stress probably persists past the time of pregnancy. It probably has a long-term effect on kind of the prevailing psychology in the region and mom's, mom and dad's psychology as well. And all kinds of other social factors are going to be interacting with this and feeding into it. So you really can't get away from stress as a factor. I guess unless you're working with animal models. In mice, you can do a lot of different stressors that are they're only happening over a specific window of time, but they tend to have a fairly large effect. Now, there are a number of effects of stress on disease risk. And to take one example, we can look at the risk of schizophrenia according to when moms were stressed. Now, one of the funny, I guess, challenges you have when you're doing this kind of work is, well, you can look at the kind of birth cohort studies if you have some very specific stressful event, or you can take these kind of cross-sectional studies and look for things that would be stressful that happen to people in a random way. There's advantages to doing it this way, because if everyone's exposed to a trauma together, say it's a famine or say it's a power outage or something along those lines, you're going to get cohort effects that kind of hang together with the whole group, and it might be difficult to tell whether you're really detecting the effect of the stress or some other kind of population stratification. However, if you choose things that are stressful and easily defined but happen in a random way in people's lives, you can kind of get around that. So here, this was a big study from Denmark that defined maternal stress as death of a close relative during the first trimester. So a close relative would be, a parent, a sibling, or a spouse, somebody sort of a first degree relative to mom. And what they find is that, first of all, fortunately, most people didn't have any stressors. So out of the several thousand that they looked at, most of them weren't exposed to anyone dying. And then if you look at when they were exposed, you can divide this up into exposed before pregnancy, sort of the antenatal period, first trimester, second trimester, and third trimester. And then you can look at the relative risk of schizophrenia among babies, or among children born to this cohort. So if you take the unexposed group as as your baseline, and say the relative risk there is 1. So somebody who is in the unexposed group of this study has as much risk of developing schizophrenia as any other random person from the population. You set that as a relative risk of 1. And then you look at the relative risk among people who were born after their mom was exposed before pregnancy or whenever else. What you will find is that the relative risk of schizophrenia is markedly increased if mom had been exposed to stress during the first trimester. So the relative risk is 1.74, or at least that's the partially adjusted one, maybe but 1.67. But you're about, you can interpret this as saying you're about 75% more likely to develop schizophrenia if your mom was stressed during the first trimester of pregnancy. Now, of course, that doesn't mean you're guaranteed to develop it. The baseline kind of frequency, or the baseline prevalence of schizophrenia is about 1%. So if you even if you were to double that risk among the first trimester stress cases, you're only talking about 2 for every 100 instead of 1 for every 100 on average. So it's not a huge increase in absolute terms, but in relative terms, it's quite a sizable increase. And it really does seem that when it comes to stress, the first trimester is the one where the biggest effects can happen. So let's take a look at some epigenetics here, now that we've defined our terms and kind of established what the concept is and the main questions. One study that by Muller and Tracy Bale looked at mice exposed to chronic stress chronic variable stress during gestation. So chronic variable stress is kind of a strategy for stressing out mice, where you go in sometimes and you rattle their cages, or you put water in their bedding, or you play loud noises, or you take urine from cats and squirt it into their cage bedding. And you basically every day you go in and mess with them in some kind of new way. This tends to stress them out quite a lot. Mice can adapt fairly well to stressors, but if you go in and do a variable, have a varied type of stress every day, then you can create some very powerful effects. And what they found was that mice who had been exposed to stress during gestation, the male ones at least, grew up to experience anhedonia and increased stress responses in adulthood. And you can measure this with a variety of mouse behavioral tests. You can look at how they respond to subsequent stressors, and you can look at cortisol levels, and you can look at how much work they would do to receive a material reward such as a treat, and these are all measures of stress and anhedonia. And anyway, these measures were all made worse by this early life stress, even though it should be pointed out these mice were not stressed once they were born. The male mice, the mice in question only experienced it while secondhand through their moms. So that's kind of a fairly well-known and fairly replicable fact about stress. But what's interesting is that this was one of the early studies to try to bring in epigenetics and DNA methylation into the mix. So what they reported was that there is decreased DNA methylation at the promoter for the CRH and the GR genes in the hypothalamus and hippocampus of these mice, respectively. So this would be what you would call a candidate gene study, where the authors of this study knew some of the genes that are involved in the stress response. You've got CRH, that's the corticotropin releasing hormone. They call it CRF here, but that's just another name for the same thing. There's GR, that's the glucocorticoid receptor. These are all genes that are involved in responding to stress via the HPA axis, the hypothalamic pituitary adrenal axis. So the theory would be that by changing the amount of these genes, or changing the degree to which these genes are expressed, would potentially change how this HPA axis works. If, for example, you had, let's say more CRH, this could mean that more CRH was being released into the bloodstream. this would lead ultimately to higher levels of cortisol and you would think more stress. On the other hand, if you had less glucocorticoid receptor in certain places, maybe that means that you're less able to shut down the stress response. There's a lot of ways you could interpret it, but the point is that they picked genes ahead of time that they knew were involved in stress. And what they found was that in the hypothalamus, the level of methylation in the promoter region of the CRH gene was reduced at a few CpGs in the stressed mice, or the mice had been exposed to stress during gestation. So here you're looking at the methylation level on the y-axis of this plot, and each set of bars represents a single CpG. Remember, CpGs are where most methylation happens. It happens at the cytosine in a CpG. So you're looking at these positions relative to the transcription start site of the gene. So these are negative numbers. That means you're looking upstream of the TSS. This is typically where the promoter, where the regulatory elements would bind and regulate gene expression. So if you have more methylation there, it might make it more difficult for transcription factors to bind, and this could cause a decrease in the expression of that gene. What they found here, though, is that the stressed mice actually had less methylation in some places. Not a lot less, but significantly less. So, here it went down from 80 to maybe 75 or so, and here it went maybe from 50 to a little bit below 40. But so, not a huge decrease, but enough to be noticed. And the theory here would be then that this gene would be expressed at a higher level in the hypothalamus because of this lower methylation. And as we know, methylation is long-lasting, it's mitotically heritable, and it can be established early in life and preserved for the rest of life. On the other hand, the glucocorticoid receptor was actually more methylated in the hippocampus of stressed mice. And if you've ever looked at this kind of stuff in a previous class, you might remember that glucocorticoid receptor stimulation in the hippocampus is pretty important for actually shutting down the stress response. It's part of this negative feedback loop that regulates stress. So this is all making a point that the regulation of the stress response can be programmed epigenetically, and that early life stress can change DNA methylation here, leading to long-term differences in how the system works. So here's another classic in the same vein. This is Weaver et al., 2004. And I'm sure you've heard of this one as well. The basic idea is this. Maternal care in rats is quite variable. They spend a lot of time licking and grooming their pups. This is part of being a mother to them. However, the amount of licking and grooming they do varies considerably between different rats. And as it turns out, the amount of licking and grooming is also heritable. It's not genetic, but it is heritable. So the amount of licking and grooming that a pup was exposed to will correlate quite strongly with the amount that that pup does when she becomes a mother. And it's not genetic because you can switch the pups around at birth, and it has more to do with what your mother did to you than what your DNA actually is. Anyway, so pups that are raised by negligent mothers therefore become poor mothers themselves. And I think if you're thinking of this from kind of a social perspective, it might occur to you that this could explain a lot of differences in human behavior as well. You're exposed as a child to a certain kind of home life, and then you carry some lessons from that with you through the rest of your life, and it might even affect how you behave as a parent. Anyhow, so back in 2004, Weaver and company raised, what they were able to show then was that pups raised with a negligent mother, a low licking and grooming mother, had higher levels of DNA methylation at the promoter for the glucocorticoid receptor gene in the hippocampus. So the part of the promoter region they were looking at was actually exon 1-7. This is actually a non-coding exon. The first 11 exons of the gene are part of the promoter region, and they're not coding, so they don't actually contribute to the protein, but they do contribute to the regulation of gene expression. They're also fairly homologous to humans, so humans have fairly similar sequences in this area. And what they found was that the higher level of methylation could lower the level of glucocorticoid receptor expression in the hippocampus, and thus, in theory, could affect the animal's ability to regulate their HPA axis, making them more prone to stress. So here's the plot of this. Here they've given the x-axis in a different way, but they've shown you the region they're talking about inside exon 1,7, and they've numbered all of the different CGs here in a little kind of a snippet of the sequence. Once you do the genome browser assignment, you'll see where you can get these sequences from, and you'd be able to find all of the CPGs yourself if you were ever curious, but there they are. And so here, for example, CPG number 15, there's a big difference. The stressed animals have very high levels of methylation. The unstressed animals in the white bars, or sorry, not stressed, but like the high licking and grooming animals with the white bars have very low levels of methylation, and that's referring to methylation at this cytosine right here, which is part of this exon. So this is something that is not genetic, because if you cross-foster the mice and switch who their mother, or sorry, the rats in and switch who their mother is, the same thing is going to happen. Now, I should point out, as is my duty, this is a very old study, and I think it probably needs to be, it desperately needs to be redone with modern methods. The fact of the matter is that methylation at this locus, measured by modern approaches, not ones from 2004, but ones from 2020, showed that this region has very low levels of methylation, being part of a CPG island. And so the numbers that they got here seem basically implausible to the modern eye. Now, it doesn't mean that the findings aren't true, but it means that the technology is old, and for the importance that this study has, to neuroscientists and people in the field, really needs to be repeated. So I'll point that out if anyone's out there who wants to repeat this. I think you do very nicely. Do this, but use whole genome bisulfite sequencing, and you'll probably find a lot of interesting things. Now, in humans, you can ask similar questions. The argument of doing rat studies in the first place is that humans might have similar things happening. However, it's very difficult to come by healthy post-mortem brain tissue, especially if you're interested in psychiatric conditions. If you're looking to study Alzheimer's, it's not terribly difficult to find brain tissue from people who died of Alzheimer's, because you're very old and you're sort of inevitable that if you have Alzheimer's, you'll die of something pretty soon. So it's easy enough to come across brain tissue from people who are sick of some kind of brain disease. Much more difficult to find healthy brain tissue from somebody who just has who has something like depression or even schizophrenia or ASD or something along those lines, because those people tend to be much younger, and so long at and typically there's no real reason why they would be at a higher risk of dying anytime soon. So it's very tough to find that brain material from them in a healthy state. What you have to wait for, and it's a little bit gruesome, is something that can give you, or something that can deliver you, a healthy brain from somebody with a mental illness would be, for example, suicide, or for example, a car accident, something that happens very quickly that doesn't affect the overall health of the brain. Because of course, if the brain is unhealthy, or maybe the person was sick with cancer or something like that, the brain is going to be different just because of that. You want to kind of capture their brain in the middle of a normal day. Anyway, so this is what they did. Labonte et al. Did a genome-wide study of DNA methylation in the brains of suicide victims with and without records of childhood abuse. So the suicide victim part is kind of incidental. That's really just the way they were able to get the brains. You need to get brains from young people who are fairly healthy, except for whatever killed them. And then you need to, and then the proxy for early life exposure to stress or to variance in maternal care was done by records of childhood abuse. So by comparing suicide victims with and without childhood abuse, you can look at a bunch of regions that are differentially methylated in the brain and make determinations about what epigenetic things might be happening there. So here is a table showing their results. So this is using a methylation microarray. So it's genome-wide, but it's not all-encompassing of every CPG in the whole genome. It only tests a very small subset of them, but in a fairly widespread way. And they found a number of genes that had differential methylation at certain positions. So most of them had more methylation in survivors of abuse, SAs. Chromosome 11, 1, 17, 17, etc. The chromosomes are given here. The p-value telling you the significance of the association is given here. The fold change in methylation, or the log fold change, is given here. So that's so all of these are increases. And then the genes are given here. So this is the gene where that particular cytosine was located nearby, and this tells you how far away the cytosine was located from the transcription start site. The logic being that the closer a methylation mark is to the TSS, the more of an influence it's going to have on expression. Although we know that because of enhancers and the three-dimensional nature of the genome, that actually things that are fairly far away can influence gene expression as well. But what I want to point out is that we've now kind of crossed the line where we're not talking about one or two genes that we can link to the glucocorticoid, or to the HPA axis that we all learned about in first year. Now we're talking about a bunch of weird genes that we've probably never heard of, like DGKZ or FHIT, and all these other ones that I wouldn't know off the top of my head what those genes do. So how on earth are you supposed to make sense of dozens of differentially methylated genes? This is where we get into a little technical point that I want to make, that this is kind of, this is sort of how I'm going to do things in this class, is whenever it comes up, I'll stop what I'm talking about and tell you about a technical side of things. So this is where we get into tests of enrichment for gene sets, and I guess you could call it like gene ontology enrichment testing. So one of the main things we deal with in the genomics age is that you're looking across the genome. The genome has tens of thousands of genes, over 20,000 probably, and you could get results for dozens, or if not hundreds or thousands of genes. How on earth do you make sense of thousands of genes and try to figure out what's actually happening at a deeper level? Well, one way to do it is to organize the genes into categories based on either a common function, like the genes all participate in a particular, a shared process, like, carbohydrate metabolism, or the citric acid cycle, or neurodevelopment, or something like that, common functions, or common locations where they're expressed. These genes all go toward the cell wall, or cell membrane. These genes all go toward synapses or dendrites or whatever. So the Gene Ontology Consortium is a large-scale project to go through all of the different genes, and for each of them, basically record as much information as researchers can possibly think of about what those genes do metabolically or in the biochemistry of the cell, where they're expressed, what processes they play a role in, and in a broader way, what other research has shown that these genes are doing. So people are actually going through, as part of this project, and going through every individual gene and going on PubMed and trying to categorize or classify all of the research that exists, even stuff that you yourself might have published, eventually might filter into this database. It's curated, meaning it's created by real humans who are literally curators. It's almost like a museum where people go in and pick out the exhibits. And ultimately, they've created this gene ontology database. And the annotations for genes, annotation is just whenever you take something about the genome and write down a fact about it, can be divided into three categories. You have biological process, which talks about large-scale physiology, things like the estrus cycle or feeding behavior. Those are the ones that I think we would tend to find most interesting in this course because they involve the kinds of things we talk about. There's also molecular function, which are cellular and enzymatic processes like phosphorylation or protein translocation and that type of thing. And then finally, as I was saying, there's also cell component, where the gene product is expressed at the cellular level. Is it in the dendrite? Is it in the nucleus? This kind of thing. So the idea here is you can feed in big lists of genes into these tools, and it will try to find common annotations that capture or that kind of describe the common patterns that those genes fit into. So instead of dealing with hundreds potentially of genes, you might find that almost all of the genes fit within a certain scheme. Like maybe all of the genes you've found are all involved in, let's say, the estrus cycle. Now you aren't talking about these genes as individuals, you're talking about them as members of a family involved in a specific process. So that's what these authors did. They used gene ontology. And by the way, this is directly addressed in the next assignment, the RNA sequencing analysis assignment, which I haven't yet released, but is mostly ready to go. So you'll be able to actually do this analysis yourself soon. But for now, I'm just telling you about it in relationship to this paper. But basically what we're talking about here is they fed in a bunch of genes that are differentially methylated in their promoter regions in the brains of in the brains of abuse survivors. And these are the names of the genes here: somatostatin, dynein, fibroblast activation, protein alpha, etc., etc. And then here on the x-axis of the heat map, you have the gene ontology terms that these genes are involved in. So if you look at this term here, cell projection and neuron projection, these are clearly terms that denote cell components, where in the cell these proteins are expressed. And that tells you that the genes that are differentially methylated by early life exposure to abuse all involve the processes of forming projections between neurons. Things like axons and dendrites are examples of projections. So it kind of narrows things down. Instead of having to think about estrogen receptor 2 beta or ER beta, or the androgen receptor, or neural cell adhesion molecule 2, or ring finger protein 6, etc. Now you're thinking, okay, we're talking about projections. We're talking about axons and dendrites. So structurally speaking, then you have a way what you have essentially is a way of linking DNA methylation caused by early life exposure to structural variation in the brain. And now if you were to go and look under the microscope and show that the dendrites looked different in these people, you would be able to link that back in with the DNA methylation and have a very nice, complete story. Does that make sense? Hopefully. Anyway, it's a way of combining this type of knowledge into a bigger schema that lets you make more sense of it. Anyway, that aside, let's get back to the real data and look at maternal infection/inflammation. So this is another one of the big things that really seem to affect your risk of mental illness later in life. Maternal infection/inflammation. So here, these authors are looking at levels of pro-inflammatory cytokines, as well as records of exposure to viruses or bacterial infection during pregnancy, and then the later risk of developing ASD, or autism spectrum disorders. So here's the table from the study. The citation is given here. And as always, when you're looking at hazard ratios or odds ratios, the baseline is 1, meaning that a group that was exposed, say, as an example, to a bacterial infection during the first trimester, their odds of developing autism, or ASD, I should say, is about the same as the regular population, 1.07, but the range is between 0.69 to 1.66, so a fairly wide confidence interval. Your odds are basically the same as any other person from the population, so it makes no difference as far as we can tell. On the other hand, exposure to a viral infection during the first trimester seems to quite considerably increase your odds of developing ASD. So the adjusted hazard ratio is 2.98. So that means you're almost threefold more likely to develop ASD if your mom was exposed to a viral infection during her first trimester. As far as we can tell, there's no data, or it's not known whether this is true for the other trimesters. There weren't enough cases. But this does fit into a pattern that we've seen, which is that the first trimester seems to be a pretty important one. And there's some other ones that are in the mix here too. However, they don't reach the threshold for statistical significance. So like respiratory infection looks like it's got a pretty big difference, but the confidence interval crosses over 0, so you can't say it's significantly different. And the same would be true for UTIs and genital infections. I mean, I guess like on average, maybe genital infections reduce your odds of developing ASD, but it's not statistically significant, so you can't say. The point is that the first trimester seems to be a very crucial time for brain development with respect to mental illnesses. So you really have to kind of watch out for infections during this time. And I think this is kind of especially relevant in our world as it stands, because we've got this viral pandemic going, and now there's just one more thing that moms can get. And it'll be a long, long time before we know if COVID infection during one of the 3 trimesters has any effect on your likelihood of, or on the child's likelihood of developing this or that. Obviously, if you want to figure out if it changes a lot the odds of schizophrenia, we're going to be waiting like 20 years, because schizophrenia doesn't tend to get diagnosed until early adulthood or late adolescence, and so we've got to wait for all these COVID babies to grow up, and we'll see what happens. So I'll meet you back here in the same place in 20 years, and we'll talk about the data. But until then, we can only speculate. Although I think I would have to say people are probably going to be researching it pretty closely because there's been a ton of money spent on COVID research, and we're going to know everything that exists about that virus pretty soon. Anyway, so something to think about. My advice would be to not get sick when you're pregnant. That seems to be the simplest thing, although easier said than done, because you know what? One of the many dimensions that causes an interaction between class and socioeconomic status and overall health is the fact that some people can stay at home and stay sheltered from all different viruses and infections while they're pregnant, and other people have to go to work and have to go deal with the public. Maybe they have to wait tables or work in an office or whatever the case may be. So, there's a real difference there that between the types of jobs people can manage to have and the health of their offspring, potentially for their entire lives. So keep in mind that we're not just talking about biology here, we're talking about our actual society and the world that we're trying to build for each other. Now, one of the things you can do instead of just looking at whether an infection was reported or not, because sometimes, a lot of times you might get sick and not even bother going to the doctor and getting a test, right? How many times have you gone to the doctor and gotten a test for what you have if you have a cough or the sniffles? Like, maybe they might test you for strep or something, but I don't think beyond that, except for COVID, you're not getting tested a whole lot. So you can use what are called retrospective markers. This is when you take a sort of a surrogate marker of a disease or of an infected state, like levels of pro-inflammatory cytokines, for example, interferon gamma, and measure them, and then without even asking what moms had, just measure the kind of the overall level of inflammation that they had at a given point during their pregnancy. So the early markers for autism study did this, and this was a case control study that looked at banked serum. So they didn't even have to go going and asking ladies for their blood. It's just that some of it happened to have been saved, and so they collected it from mid-gestation women who gave birth either to a child who was diagnosed with ASD or not. So they had tons of data, and what they did was they measured a couple of different cytokines, interferon gamma, interleukin 4, and interleukin 5, and compared the levels in moms who had given birth to a child who had ASD to moms who had given birth to a child who did not have ASD. And what they found was that the levels of mothers of ASD babies or AU babies had higher levels of interferon gamma than control mothers at mid-gestation, suggesting that they were under a higher level of inflammatory stress, potentially arguing that maternal inflammation affected brain development in such a way as to predispose to autism or autism spectrum disorders. This is higher than the general population and also higher than mothers who gave birth to babies with other kinds of developmental disabilities. So this is a way of getting at this question without ever actually going and talking to anybody or doing any diagnostic tests on them. Now, you can kind of go on and on with this, and this type of stuff is fairly well characterized and fairly well studied, although very little has been made, very little about it has been made conclusive, because it's very tough to take these facts and translate them into anything that you can do other than the broad advice that you should try to be as healthy as you can if you're pregnant, which is easier said than done for a lot of people. But you know what? Developmental programming doesn't only have to be negative. It also can be about positive outcomes, because as bad as the environment can be, you can also have very nice environments that actually make things better in a long-term way. So trying to bring some positivity into the mix, Zoker and company back in 2020, that great year we all remember and love, showed that early life environmental enrichment, which in this case was building a super cage for mice. This is a mouse study, and the super cage is essentially a couple dozen cages all connected to each other, so the mice can all be part of a big town. They can go visit each other's cages, and they can hang out and have friends and explore and do all kinds of things that are much closer to what a mouse actually wants to be doing and what a mouse is actually supposed to be doing. What they showed is that exposure to this type of environmental enrichment led to greater levels of what we call behavioral individualization in mice. So if you think about stereotyped behavior as a mouse doing the same exact thing over and over again, that's what a stereotype is, right? Doing the same repetitive thing. Behavioral individualization is the exact opposite of that. Mice behave in a very kind of almost creative way where they do new things all the time. They're, the difference between a boring pet and an interesting pet, I guess. And the enriched environment leads to higher levels of that. And naturally, I guess you could imagine, if you know a few things about the brain, this also led to increases in hippocampal neurogenesis, because most of the good things you can do to mice do cause neurogenesis in the hippocampus to increase. And the behavioral changes persisted even after the mice were removed from this enriched environment and taken to an impoverished environment. And so you can see the persistence. So this is a picture of the super cage up here. It's all just a bunch of cages connected together, and it's a pretty simple way of doing it. They do this because these cages, the labs are built with cage washers, so you can wash these cages really well, and it doesn't require a whole lot of special time and effort on the part of your animal care team. And this just shows the stability. So the amount of novel object exploration during the first phase plotted against the novel object exploration on the second phase. So let me just take a quick break here, and I'll be back in a few minutes to tell you about the results of this study. Okay, and we're back. So let's get into the results of this study. They looked at, obviously the enrichment itself is quite interesting, but they also did some epigenetic analysis. So we can take our newfound knowledge of DNA methylation and apply it to the results from this study. So it doesn't really matter how they measured it, but we'll just tell you about the results. So they looked at genes that had differential methylation, meaning the amount of DNA methylation at these genes, or more accurately, the promoters of these genes, was significantly different, either higher or lower in one of the in the enriched group compared to the control group. And so this Venn diagram here, what it shows is that for mice exposed to the enrichment, all of the genes that would occupy this circle, so 5800 plus 3200, whatever that number is, that's how many were differentially methylated by the enrichment, either had a change for the increase or a decrease. And here are the number that were differentially methylated when the mice were enriched and then switched to a standard cage for a while. So these are the ones that are kind of persisting. The ones in the middle part of the circle, 3200, these are genes that are differentially methylated by exposure to enrichment. And they remain differentially methylated in the same way, even if you take the mice that were once in the happy cages and put them in the bad cages. The bad cages are about the size of a shoebox, and it's a very boring, bad life for a mouse, but that's, by the way, how most lab mice live. So there were over 3,000 genes that had persistent differences in DNA methylation in response to this early life enrichment that enabled them, or at least theoretically, correlated with their tendency toward exhibiting more complex and more intelligent kinds of behaviors, more behavioral individualization. Now, if you look at the gene ontology terms that are linked to these 3,200 genes, the pathways that come up have to do with the ephrin receptor signaling pathway. So the ephrin signaling pathway it's a pathway involved in neural development and it guides the migration of neurons and so on and so forth. So it's a gene that you would expect perhaps, or it's a set of genes, I guess, that you'd expect to be increased in mice with a lot more brain development happening. And, in a way, that's sort of what we see, right? Because there's more neurogenesis in the mice that were exposed to enrichment. They have more complex forms of behavior, and you would think perhaps that means they have more highly developed kind of, brains, whatever that would mean. But anyway, this is reflected by differential methylation in ephrin receptor signaling pathway genes. And these are long-lasting. For example, if you look at the ephrin B1 gene, so here's a gene browser or a genome browser kind of view of the gene. You've seen this kind of diagram before, right? You've got the introns and the exons here and so on and so on. And the area that they looked at, the CPG island, is right here. And what they find is that at these positions, the gene is on chromosome X. These numbers here give the position on the X chromosome. So think back to your genome browser assignment. Every position is given a number that just tells where it is on the chromosome from 0 all the way to the end of the chromosome. The methylation level here is much higher in mice exposed to enrichment, whether or not they've been switched back to a standard cage. So in this case, the enrichment has a long-lasting impact on methylation at this particular gene. This is one of those things where it's also a big enough change that it makes me kind of skeptical, actually. Normally you don't see changes this big. But anyway, this is what the authors reported. So it's good news in a way, right? I mean, we're trying to make a point that, like, as bad as it is to have famine and stress and all the rest of it, the good things we do in society that make life better for moms and for children can have positive effects that are equally as persistent and long-lasting. So that's probably what we should be trying to aim for, don't you think? Now, there's a concept called transgenerational effects. This ties in with the broader notion of generational trauma, which is the tendency for traumatic experiences in one generation, poverty, war, famine, abuse, exposure to residential schools, concentration camps, etc., to impact the mental and physical health of subsequent generations, even though they themselves were not directly exposed. So there's more than enough evidence that this is a real thing, and it can be transmitted through behavioral and psychological changes and cultural changes that pass through the generations. You don't need any biology at all. I should be very clear here. This is real whether or not the biology is there, because it happens at a psychological and cultural level that we can observe quite plainly with our own eyes. However, there does seem to be growing evidence that in addition to this, there is some level of epigenetic transmission as well. And I guess, I wrote here that just because we have some epigenetics in the mix, really shouldn't take away from the social and psychological aspects of it, because there's kind of a risk, I think, of pathologizing this to say oh, well, the reason this is happening is because your DNA methylation is messed up. And it's well, that's not really, that's just sort of an effect. That's not the cause of this. The cause is the actual trauma itself, whether it's, war or concentration camps or residential schools. It's not a medical matter. It's a matter of oppression, and oppression that could be either happening now or in the past or both. So I just want to stipulate that so we don't try to medicalize this away. Trauma that is endured by the pregnant mother, for example, doesn't affect just her and her fetus. Well, it does affect her and her fetus. That's pretty obvious, right? But it also affects her grandchildren, because while mom is pregnant, the primordial germ cells that will give rise to either the sperm or the egg of her offspring are also in there growing it as well. This is especially relevant to growing females because, of course, males are, sperm in males, that's continually being turned over, and it doesn't, you're not going to have the same sperm cells this day as you will next week. Whereas for the eggs, those are made during fetal development, and they stay there the whole time. So whatever they're exposed to can be quite long-lasting. And this is sort of gets into the idea that maternal transmission of trauma and whatnot is easier to understand or to imagine than paternal transmission, because the fetus lives inside of mom. So whatever mom is exposed to, the fetus is exposed to as well. If dad is stressed, it's just harder to imagine how that stress would be transmitted to the offspring. The only way it could be, it would have to be somehow packaged into the sperm and carried into the into mom, and you know how that whole thing works. And it's possible, it seems like it's possible, but it's just mechanistically a great deal tougher to understand, because you're not, it's not like the male's there. I mean, a lot of, the dad needs to be there basically for about, 2 seconds, and that's all it really required to lead to conception and pregnancy and everything. Now, but even in both cases, the epigenetics are a little bit confusing, because as I mentioned in the lecture on epigenetics, DNA methylation is erased during meiosis, so almost all of the methylation that you could plausibly have in there is erased. So how could it be transmitted into the germ cells? So, this has been looked at a fair bit, and I think the issue is still fairly controversial, but I'll give you one example of a study that was done about a decade ago now. So in 2011, Dunn and Bale conducted a transgenerational study of maternal high-fat exposure. And in a nutshell, they took F0, the first generation, and they fed these moms a high-fat diet before and during pregnancy to make them overweight. And then they took the offspring of those and mated them to control mice that were not exposed to the high-fat diet. And they ran this all the way out to F3. So these mice here would have absolutely no direct contact to the maternal obesity here. These mice would be because they were in utero during the treatment. These mice would be because they weren't in utero, but the germ cells that gave rise to them were located inside of these mice who were in utero. So F1 and F2 are both exposed, but F3 is completely separate. So if there's any lingering effects on the metabolism of F3, it would have to be through some mechanism other than direct exposure to the treatment that they gave to the first generation. Does that make sense? What they found was that F3 mice, meaning mice that were all the way over here and not directly exposed to any of this, still had, in the paternal line I should say, so F3 mice that had a dad who had been exposed, not a mom, F3 mice that had a dad that had been exposed to that maternal obesity back in the earliest generation showed elevated body weight relative to controls. Now, it's not a big difference, but it's statistically significant, and that's good enough for us. So this would suggest that some kind of change, potentially of the epigenetic variety, has managed to be transmitted all the way to F3, taking a sort of a detour through the paternal line, which is really quite a implausible, or it's a pretty bold claim if you think about it, because here's the mom who's exposed. She, anyone who's inside of her uterus is going to experience this and get exposed as well. So let's say it's the males. The males are going to be exposed. They themselves will be at a higher risk of obesity. This is no big surprise. Then when they are mated to control mice, the control mice are once again have some level of residual exposure because they were the germ cells of this male had been exposed. So there's still some level of contact there. But then by the time you get to F3, all of that theoretically should be washed out. And yet the fact that by F3, female mice exposed to maternal obesity by the paternal lineage still have elevated body weight. Now, this is a pretty, that's kind of an open-ended question. Why would they have that? Well, the authors of this paper, this was 2011. The technology was not really super advanced in those days. So what they ended up looking at were the expression of what are called imprinted genes. So this is just one of the worst, I have to say, really one of the worst looking figures that's ever been published in a paper. And I apologize to Tracy Bale and her colleagues. They do good work, but holy Lord, that's a terrible figure. It's a three-dimensional bar graph, and it's essentially showing the differential expression of paternally expressed and maternally expressed genes relative to controls. The height of the bar represents the amount of differential expression. So the really high bars might be 1.5-fold higher in the obese lineage and whatnot, but could there ever be anything harder to read? And also, it doesn't tell you what the names of the genes are. You have to look them up by saying, oh, what's gene A8? And there's a table in the paper that tells you. So please never make a figure like this. Make a heat map or something else. This is just really awful to look at. So there's not much we can really say about those results, but there were some differences in these imprinted genes. Now, what is an imprinted gene, you might be asking? Well, this is another one of these notes that we're going to put in the side. This is kind of how things are going to work from now on. Now that we've gotten past the main introductory sections, is I'll mention something that a paper talks about and then tell you what that means. So, do you recall our discussion of exon activation? What it means, and we just talked about it last week, so I would hope that you do. But exon activation means that for any given somatic cell, only one of the, in genetic females, or with a female sex with two X chromosomes, only one of the two X chromosomes is ever going to be expressed. The other is completely silenced, for the most part. This is what you would call, so this is X inactivation, but it's a special case of a more broad concept called monoallelic expression. Mono meaning one, and allele meaning allele. So you have two alleles for every gene. You have one that you got from your mom and one you got from your dad. And typically, you would express these biallelically. You'd express mom's copy and dad's copy in about equal proportions because your autosomes, there's no X inactivation for chromosome 21, for example. They're both expressed in equal amounts, and they both presumably would contribute about equally to the proteins. This, by the way, is how this is why having a recessive allele. This is how having a recessive allele works. If you have a bad version of the gene, you still have a good version of the gene from your other parent that's being expressed and is working fine. So for biallelic genes, you've got two copies that are being expressed roughly equal amounts. Monoallelic expression, which would be an example, like X inactivation would be an example, but there are others as well. That's when only one of the two copies you have is expressed. And so imprinted genes are a type of monoallelic expression. Imprinted genes are when either the maternal or paternal copy is epigenetically silenced by DNA methylation. So you only express one or the other. And these are not on the X chromosome. Very important to say. These are on any other, they could be on any chromosome. So in the case of maternally imprinted genes, only the maternal allele is silenced, and all of your cells will only express the paternal allele. You'll only express dad's copy. And for paternally imprinted genes, it's the exact opposite. So these are a very small subset of genes where, unlike most genes, you only express one copy, either from your mom or your dad. And it turns out that if you look at these genes, and again, there's only about 24 of them maybe, they have some very interesting phenotypes that they're related to. So the maternally expressed genes are given here. One of them is GNAS. This tends to reduce the levels of locomotor activity in mice, and it also reduces the level of growth. But it stimulates the amount of maternal demand, so how much a baby mouse needs its mom. On the other hand, the paternally expressed genes tend to increase the amount of suckling. They tend to also really increase the amount of growth. So the genes that dad gives you are all, and that are being expressed only from your dad's copies, are all concerned with making you consume more of your mom's resources, essentially. They lead to more suckling, they lead to more growth, they lead to higher metabolic rates and body temperature. They basically make you bigger and stronger. And so there's kind of a bit of an opposite thing there, where maternally expressed genes tend to reduce growth and paternally expressed ones tend to increase growth. And you might ask, well, why would this ever happen, right? It's kind of a weird phenomenon. It doesn't, you would think that why wouldn't you have biallelic expression for everything? Well, we're not really sure why, but a few people have come up with reasons. Wilkins and Haig are two, and they basically argued this. Moms tend to invest more resources into their offspring than dads because a male animal really doesn't have to do any investment at all. A male animal can really just, mate and then disappear forever. This is, fairly common in nature. Whereas moms have to contribute all of the nutrients and all of the resources and all of the caretaking. If if a mom wants her genes to be passed on, she has to do a ton of work. Dad really doesn't have to do a whole lot. It depends on the species and it depends on the dad. But, broadly speaking, this is a sort of a fact of biology. So this leads to a conflict in resources or over resources about what moms and dads want. Dads, evolutionarily speaking, to pass their genes on to efficiently pass on their genes, kind of want the baby to consume as much of mom's resources as possible because the baby has dad's genes, and there's no downside from the point of the dad if the baby, consumes everything the mom has to offer, especially if it involves outcompeting the other siblings that may not have the same dad. So dads just, they kind of just want the baby to go to town, whereas moms would want to do the opposite. They want to conserve their resources so they can be spread out and, managed over all of the mom's brood or all of her offspring and, over her entire life, right? She has a future ahead of her. The dad may not care about this. So many of these imprinted genes are expressed in the placenta, and typically the ones where you express dad's copy all lead to higher growth rates and higher demands on the mom, and the ones that that are expressed from your maternal copy do the opposite. So this is a theory, but it's only a thought. We don't really know why. But it is important to point out that this is controlled by DNA methylation. So the way this works, and here's another gene diagram. So let's take PEG3. This is a gene that in mice is paternally expressed, meaning it's maternally imprinted. What you will see is that on the chromosome, or the near the allele that you got from your mom, on the piece of the chromosome that you got from your mom, the transcription start site and promoter region of this gene will have a lot of DNA methylation. These closed circles represent methylated cytosines. So there's a lot of methylation upstream of this gene. So that would prevent the maternal copy from being expressed, because DNA methylation typically leads to less expression. On the paternal allele, on the other hand, you have very low levels of methylation upstream of this gene, meaning that that particular copy is expressed. The methylation takes place at these things called ICRs. These are imprinting control regions. So the methylation level there correlates with which chromosome that particular allele came from, to the point where you can actually look at the methylation and say, oh, this came from mom or this came from dad, if you know what gene you're looking at. For imprinted genes, not for every gene. And like I said, there's only a few dozen imprinted genes. And they're often targets for developmental programming because they regulate so many important aspects of growth and development. And because imprinting is something that's epigenetic, it's known to be epigenetic, you would think that it's more labile, let's say, during development, because epigenetics can change. So some disorders, by the way, like Prader-Willi syndrome and Angelman syndrome, are genetic disorders that involve imprinted genes. So Prader-Willi involves a paternally expressed gene, and Angelman involves a maternally expressed gene. So we don't need to really remember that too much, but it's an important point to say that for a very small subset of genes, you express them monoallelically, either expressing just your mom's copy or just your dad's copy. So paternal transmission, as I've said, is a little harder to understand. It's easy to kind of imagine how developmental programming works for maternal transmission, because you're literally in your mom's womb while she's experiencing an infection or a stressor or something along those lines. The dad, it's not quite clear. I mean, like I said, males don't need to be too involved with reproduction at all. Male mice can come in, mate for 5 seconds, and then leave. And it's very tough to imagine how much they really would be able to communicate to their offspring about stress and about other developmental things of that sort. So methylation, for example, is not going to be transmitted through the sperm, because it's mostly erased during spermatogenesis. So if you're trying to imagine a mode of paternal transmission where the stress that dad experienced in life is being transmitted to the offspring, and dad's not in the picture, he's gone, you need something other than DNA methylation to carry that information. And so one suggestion has been microRNAs. MicroRNAs are really small fragments of RNA. They were discovered, I think, they were discovered maybe in the last 15, 20 years, comparatively recently for an old person such as myself. But they are little fragments of RNA that are able to bind to mRNAs, messenger RNAs, and block them from being translated. And so they're a way that cells can fine-tune gene expression. I mean, the cells have a million ways of fine-tuning gene expression. MicroRNAs are just one of those. And anyway, so in 2014, Gap and company showed that mice that were exposed to early life maternal separation, this is a very good way to stress out newborn mice, is by taking them away from their mom for a little while, and unpredictable maternal stress. So you take the pups away and then you stress the mom out by shaking her cage and doing all kinds of horrible things. Those pups grew up to have depressive-like behaviors in adulthood. And importantly, these were also seen in the offspring of males. So you were looking several generations ahead and finding that even by the time you're at F2, the offspring of F2 still have some differences in various kinds of behavioral traits that are linked to stress. So they're elevated plus maze, or they're light dark box, and these kinds of various tests of rodent stress. So there is some aspect of this experience that occurred all the way back in F0 that is transmitted across the generations on the paternal line. You can show this, so something's got to be getting transmitted. But what is it? Well, in this case, the researchers looked at microRNAs and found that a number of them were elevated in the F1 sperm cells. So this is them. I don't want you to worry about what these do or what they're for or anything like that. MicroRNAs are kind of not really are in our, they're not really part of this course exactly, because I don't really consider them to be a type of epigenetics, but they're there and they might explain this, because if you can, it's one more type of information aside from DNA that the sperm can carry along with it. And so we're going to, so we'll just leave it there. There's a lot more to be asked about this and verified before we can really say for sure that this is how it works. But in addition to the DNA, let me just put it this way. In addition to the DNA, there's also epigenetic information as well as microRNAs that all travel along in the sperm and in the eggs. And any of these three things can explain differences in phenotype. And if you take away DNA, both epigenetics and microRNAs and things of that sort can explain inherited phenotype, or can explain acquired phenotypes, I should say. So stress, susceptibility, and so on. So it's a possibility, but it requires a fair bit more work to really decide on whether it's true or not. So I'll end on this slide here. What I'm trying to communicate is that developmental programming can happen within the same, within nearby generations, from mom to daughter, and it can also happen transgenerationally from mom to from mom to grandchild, or from mom to great grandchild, etc. It's an important part of understanding disease risk, because we can only go so far with genetics. Even the world's most highly genetic conditions seem to have a fairly large component of the risk that's not genetic. And what's good about this is that we can actually change the environment. So we're not going to go out there and change anybody's DNA anytime soon, but we can probably change the environment. We can ask questions about how much of our capacity is innate and say, well, the things that we like, let's try to build an environment that makes those things more common and more likely. I like the idea of investing in maternal health and early childhood because the return on investment is tremendous. You can do some pretty minor changes to the welfare system to massively improve the situation for moms, and the effects on health will be the last through the entire lifespan. So it's it's kind of baffling on on a purely economic level why that's not why people don't pay more attention to that. Although on a basic human rights level, it should also be looked at. Anyway, so I just want to bring that up as a some of the amazing potential of this field. But not to make you focus only on the bad side, I suppose. The field isn't perfect though, and I think I have, this is kind of my field. Maybe this is why I'm continuing to rant even a few minutes after the class is technically over. It's not perfect. Probably most of the work is of low or questionable quality in mice, and I really think that we don't, it needs to be kind of modernized and brought up to a modern standard. A lot of it's a little bit old and just kind of outdated technologically. So we need to bring this up to date, and we need to see how this extends to human beings. A lot of these models are very mouse specific, and we have no clue if they would ever happen in humans or not. So that has to be done. And so I think that's where we'll leave it, folks. There's a lot of pitfalls with this, but there's a lot of potential as well. So keep that in mind. We're going to be talking about this a little bit as we go through and look at different diseases. So I'll leave it there, my friends. Thank you very much. So on our next class, we will be having a bit of a lecture where we talk about the ways in which we can use genomics technology to link genes to phenotypes. Basically, we'll be talking about GWAS studies and sort of their relatives in the field. And then we're going to go into, I think we're going to do a bit of a review. I don't want to talk the whole time about new material. I want to just go back and maybe revisit some of the concepts that I think could have been confusing, or maybe that I didn't explain very well, or might have been so new to you that you would want to hear it a second time. So I'll go and review a few things that I've chosen. And then, of course, I would, of all the lectures to visit in real life, I think this would be a good one. I mean, to visit live, to come to the live stream for this one, because you can ask questions and I'll be able to answer them. And like I say, there's a quiz, a pretty valuable midterm coming up a week from today. So you do want to look into getting the help you need if you need any. So folks, I'll leave it there. Thanks to everyone for listening, and hope you have a good week.